In Chapter 2, we'll talk about the physical properties that basically will drive the anatomy and physiology of a cell, focusing mostly on chemical bonds, which I'm sure you've seen quite a few times before. We'll focus on the second law of thermodynamics and the role of ATP in cell physiology, or adenosine triphosphate. And finally, on the last slide, I'll go over the individual definitions that relate to Gibbs free energy, entropy, and enthalpy, and as well as go over one particular equation that helps us figure out whether it is an endergonic or exergonic reactions, which again, something you should be familiar with in previous classes. Now, covalent bonds are the strongest bonds that we know of with molecular structures. And every single molecule is looking to fill out its outermost valence electron shell. Once it fills all positions of the valence shell, it becomes a lot more stable as far as the molecule goes. In this case, we look at carbon. Carbon is the most well-known atom that we have, especially in organic chemistry. As you can see, carbon has a 4-electron outermost shell, so it can accept 4 other electrons and fill up to 8 to give the maximum occupancy of the valence shell. In this case, we see 4 hydrogen atoms that will share electrons with the carbon to form methane. We'll also talk about how the number of bonds between 2 particular atoms can allow certain things to either fully rotate or whether they stay relatively rigid. Or we'll also look at different types of covalent bonds that either achieve either a non-polarity or polarity in certain types of molecules. Going back to methane, we'll look here as we revisit the polarity or non-polarity of certain types of molecules. So when we see an even sharing of the electrons, both in hydrogen, each hydrogen to the carbon atom, these electrons are shared equally between the individual atoms. So there's no net charge. So in this case, methane itself is known as non-polar, which means that there's no area where the electrons are going to be favored around one particular atom versus another. So it's all equally shared. So again, there's no polarity here. We get into different types of aqueous reactions. We'll talk about how they will manifest themselves within the aqueous solution, whether they react with water or not. In this case, methane being non-polar will actually not associate with the water. In fact, it will pull itself together. Next, we have the single versus double bonds, and in some cases triple bonds, depending on different types of atoms involved. In this case, we look at ethane on the left. We see the single bond between 2 carbon atoms, as each one is going to be filled. Otherwise, with hydrogen atoms, it allows free rotation along the bond of one carbon on the other. However, if we add a second bond between 2 carbons, basically they'll form more a rigid bond as we go from ethane to ethene. This is a very rigid structure, which we're going to look at a little more closely when we get into hydrocarbon tails. In particular, when we look at phospholipids and triglycerides, we start looking at different types of lipids we find in and along the outside portions of the cell. Revisiting polarity and non-polarity, this should seem very familiar to you. This is a water molecule. In this case, with the covalent bonds, we see there is a non-equal sharing of the electrons. They tend to hang out a little more so along the oxygen atom versus the hydrogen atoms. This gives a polar molecule, so that's polar opposite ends, one being negative and one showing positive. So obviously, the oxygen atom would be more negative, and 2 hydrogen atoms on the opposite sides would be more of a positively charged area. So this actually helps us with different types of polar molecules where they will actually start to dissociate in water, which is very important for different types of reactions that we find both in and out of the cell. Covalent bonds play a very major role in different types of chemical reactions found within the cell. So basically you see this particular chemical reaction, you have certain types of reactants on the left, 2 amino acid glycines that will turn into a product of diglycine and water. In this case, going from left to right, this is what's known as a dehydration synthesis, where water will be one of the products and you're joining 2 distinct amino acids into one. In this particular covalent bond, this is known as a peptide bond because you're joining 2 amino acids. Now, as is most reactions found within the cell, this is actually reversible. So going from the opposite direction, your reactants will now be diglycine and water. And then going from right to left, you'll have 2 reactants, or 2 products, amino acid glycine. This is what's known as a hydrolysis type of reaction. So again, in the cell, we're going to be talking about quite a bit of covalent bonds being both formed and broken in different types of different reactions. Ionic bonds tend to be weaker than covalent bonds, so because with the ionic bonds, we don't have a sharing of the electrons, we see a donating of electrons from one atom to another to form an ionic bond and form a particular type of molecule. So we go back to the most common ionic bond explanation or example that we see here. We have sodium and a chloride atom. So again, if we're trying to achieve a full valence shell, the sodium only has one electron in its outermost valence shell, whereas the chlorine atom can accept only one in its most outermost valence shell. The sodium ion, instead of sharing the electron, will donate to the chlorine atom. So what happens is the sodium atom will become positively charged, known as a cation. So those of you who need help remembering, if you say you're positive it's a cat, you'll remember that cations will be positively charged atoms. So what that does very simply is it oxidizes the sodium atom because it loses an electron, which therefore reduces the chlorine atom and therefore becomes more of a negatively charged or anion atom. And then this ionic attraction between the positive and negative poles on both the sodium and chlorine atoms will form the table salt molecule. Now, one thing to remember too is the sodium atom becomes a reducing agent because it reduces the chlorine atom. And conversely, the chlorine atom becomes an oxidizing agent because by accepting the electron, it oxidizes the sodium. Now, I'm sure most of you have done this experiment before. If not in the lab, I'm sure you've done it at home. Taking crystals of salt or sodium chloride, again, knowing that you have both positive and negatively charged atoms within the molecule. If you were to drop them in aqueous solution, in this case in water, it would simply dissolve the salt molecules and break down into individual sodium and chloride ions. So because of the polarity of the water molecules, the negatively charged ends of the oxygen will attract themselves and surround almost into an aqueous shell around the sodium ion, whereas the hydrogen or the positively charged ions will be attracted to the chloride atom to very simply form a shell around there as well. Now, if you extrapolate this further, if we were to take a nonpolar molecule and drop it into water, it would not dissolve into water, as there would be no reactions between the polar molecules of water. So there would be nothing for it to react to on a nonpolar molecule, so it would disassociate in water and form droplets. In this case, if you think of fats or lipids, they form oil droplets within the water, and it doesn't dissolve completely. Now, hydrogen bonds are going to be the weakest bonds that we have, but in high quantities, they actually make things very, very stable. But because of the easily broken hydrogen bonds, this is very important for certain types of reactions that we see all throughout the cell. In particular, if you look at certain types of reactions where there's a change in pH, if you remember there's either too many hydrogen atoms or too many hydroxide atoms, that pH can damage the hydrogen bonds and therefore break things down. So in particular, when we look at certain organisms that have stomachs with hydrochloric acid, this is very important to help break things down very easily. Now, fortunately, these are also very easily formed. We start talking about different types of amino acids that they start to form into different structures for proteins, enzymes, and so on and so forth. These multitude of hydrogen bonds are very important to maintain that structure, but also that easily breakable bonds. Very important for different types of enzymatic reactions, both in the creation of products as far as anabolism or catabolism, we're trying to break certain structures down. Now, shown here, different hydrogen bonds can be formed either between individual water molecules or oftentimes between water and different types of organic molecules. This will come into play quite a bit. We look at protein synthesis as well as looking at both the zipping and unzipping of DNA for both for transcription as well as duplication of the DNA. Hydrogen bonds will also play a major role in different types of enzymatic reactions as we look at different types of sites within the enzyme itself to be turned both on and off with allosteric sites as well as inhibitory sites, as well as the active site where certain types of substrates can be changed into different types of products. The easily broken hydrogen bond or reforming hydrogen bond allows these particular enzymatic reactions to occur with relative ease. Now, we talk about different types of reactions with water or any kind of polar or nonpolar molecules. We always talk about either hydrophilic or hydrophobic or amphipathic types of reactions. So hydrophilic very simply means water loving. In other words, they will be attracted to either end of the water molecules. In this case, on the left, we see a polar molecule that's going to be attracted on either end. It looks like on the left-hand side, you see the green portion will be negative, so it's going to be attracted to the positively charged poles of the water molecule here. Whereas conversely, the whiter side of the polar molecule is going to be more positive, can be attracted to more of the oxygen portion of the water molecule. If there is no polarity found in the molecules, they will not react with individual water molecules and oftentimes pull together. So we look at different types of macromolecules, we look at different types of proteins and how they fold. Usually, if they're going to be in aqueous solution, the hydrophilic portions will actually face outward towards the water, whereas the hydrophobic portions tend to be more towards the inner portion of the protein. And we talk about other types of molecules, we're talking about different types of lipids, the plasma membrane, triglycerides. This also comes into play as far as how easily certain types of reactions can occur either in the cell or sometimes in interstitial fluid or sometimes on the aqueous surface of different types of organisms. So again, just a closer look. So again, polar molecules will have a hydrophilic reaction with water, means that they're water loving. We do have some molecules that are known to be nonpolar. These are known as hydrophobic, which means they're water hating. So they will not react with the surrounding water molecules. In fact, they will actually start pulling themselves together. Oftentimes, we will see certain types of molecules that will be both hydrophilic and hydrophobic in different sections. So we see different types of phospholipids and proteins. So these are known as amphipathic type of molecules, which have some portions hydrophilic versus hydrophobic. So will fold in a certain position to either interact with different types of water molecules or try and shy away from water molecules. Now, the last type of interaction we'll talk about as well are known as van der Waals interactions. These are the weakest of the four types of bonds that we see among molecules. These occur more so in nonpolar molecules, not so much with polar, because the ionic bonds and hydrogen bonds tend to override most of your van der Waals reactions. However, under nonpolar molecules, because of the split-second unequal sharing of electrons in any given moment, a certain electron may be spinning around one particular atom and spinning around another. Occasionally, you'll get what's known as the van der Waals reactions that you see, where everything will line up in a certain position. The gecko that we had looked at back in Biology 1 oftentimes has the van der Waals interactions with certain types of molecules, which by themselves are relatively weak, but when you look at increased surface area of certain types of molecules and so on and so forth as they interact with different types of surfaces, occasionally the van der Waals reactions can interact very cleanly, and in greater numbers they actually have a very high attractive force. Here's the adenosine triphosphate, which is the major cell store of energy. So if you look at the actual structure of the ATP, if you look, it has 3 phosphate groups, and the most energy is stored between the second and third phosphate group, although you can derive other energy from the first and second bond of that particular phosphate group. You see this attached to a ribose sugar. It's a 5-carbon sugar, and therefore also attached to the adenine nucleic acid. So very simply, this does share some characteristics as DNA molecules, with the exception of the additional 2 phosphate groups along the triphosphate. So as we start to break down between the second and third phosphate groups, this releases quite a bit of free energy. So it decreases free energy from the ATP to release to other cellular functions to be used as the cell sees fit. You do get some energy that will be derived from the adenodiphosphate into the AMP, but it's not quite as high as what you see from the ATP to ADP, mainly because there's a higher concentration of ATP, adenosine triphosphate, than adenosine diphosphate found within the cell. So we get a better yield from ATP to ADP than we get from ATP down to AMP. So let's round out this section talking about different types of terms for energy that we use within the cell. One of the terms that we use, known as delta S for entropy, is the degree of disorder in an isolated system that increases over time. Now we know the cells are not isolated systems because they're constantly being shunted in with different types of molecules that can generate ATP, which includes carbohydrates, different types of proteins, as well as lipids. So even though the cell is constantly churning out waste products and heat and increasing entropy, it's constantly replenished by additional energy stores. Now, one of the things we talked about with the ATP hydrolysis, we talked about free energy. So this is known as Gibbs free energy, therefore known as delta G. So it's the energy that's within the system that can be used to do work. So in this case, when the ATP is broken down to ADP, that free energy is released from that molecule, but it's going to be used for a different reaction somewhere else within the cell. Oftentimes, this is measured either in joules or in calories, or more oftentimes in kilocalories, which you'll see oftentimes on the side of a nutritional panel of a particular type of foodstuffs. The other type of energy we talk about is enthalpy, which is delta H. That's the heat released or absorbed during a chemical reaction. This, if you remember, helps us give an idea of whether we have either an endergonic or exergonic reactions. Typically, endergonic reactions where the free energy is going to be greater than 0 requires free energy input. So there's energy investment there. Whereas an exergonic reaction tends to have a free energy output, so it's lost. So that tends to have a release of free energy, oftentimes to a different type of reaction. So the equation that we use from time to time is going to be delta G, or Gibbs free energy, equals the change in enthalpy minus the absolute temperature times the actual degree of entropy. So very soon that gives an idea of prediction of whether a particular type of reaction will have either an endergonic or exergonic reaction. So an exergonic reaction can actually happen instantaneously, whereas an endergonic reaction needs some kind of energy investment, oftentimes to create a certain type of product. Usually exergonic reactions tend to break down certain types of molecules into smaller molecules, whereas endergonic reactions tend to be more of a synthesized reaction where you're taking smaller molecules and building up something larger. Say in the human body, let's say we're trying to build up muscle, we're trying to build up new skin tissue, those would be more endergonic reactions requiring energy input. Again, in later chapters, we'll talk about how different reactions happen within the cell, where you're constantly starting to break down or catabolize certain types of molecules while you're conversely trying to build up or anabolically build other types of molecules. So this finishes up this particular section. We'll move on to 2.2. We'll talk about different types of reactions and how things are being used within the cell.